So in today's lesson, we have two tasks to accomplish in finishing up the light reaction portion of photosynthesis. We're going to first talk about chemiosmosis as a follow-up to non-cyclic electron flow. And then we're also going to have to talk about cyclic electron flow. And by talking about those two topics, we'll bring an end to our conversation around the light reactions and move on to the Calvin cycle. But before we begin today, what I want you to do is, you'll notice that I've listed all of the components from non-cyclic electron flow, all here. And what I want you to do is take a minute to try and organize those, the names of these molecules and place them in the correct sequence according to how they're introduced into the non-cyclic electron flow. Okay, so I am going to go ahead and do this. So let me see. I'm going to see if this works here. So I'm going to make a copy of this molecule. I know that we start with P680. And then once P680 hits, then I'm going to move on and promote an electron to my primary electron acceptor, which for photosystem 2 is PQ. So once the electron from P680 has gone to PQ, it then gets transferred to our protein complex. This is the B6F complex. So that's going to come next. And I'm running out of room here, so I'm going to write my little arrow down here. So once B6F comes into play, then I transfer the electron to another mobile carrier. That mobile carrier's name is PC. Once that's done, then the electron goes from PC to filling the hole in, you guessed it, P700. I should probably do this. Here we go. Boom, boom, boom. So then once we fill the hole in P700, where is that electron going? Well, that electron is being transferred to ferredoxin, which we've simplified as FD. FD then transfers its electron to NADPH reductase, which, as you can tell from its name, is an enzyme. And finally, in my list, the only thing left is NADPH. So there we have it. I'm hoping you did okay with the sequence of events. There are some topics that we didn't really talk about in just outlining this, though, that I think are really important. So I want to just spend a couple of minutes talking about that as well. The first of which is, well, what are the two important products that result from non-cyclic electron flow? So I'm hoping that you would have said ATP and also NADPH. And so, well, what we haven't talked about, we've talked about NADPH as the final product of that electron transport chain, but we've only sort of touched on ATP. And so what I'm hoping that you're remembering from yesterday is that, and this is meant to depict a thylakoid disc, and all of the components of our electron transport chain are embedded in this thylakoid membrane. But at certain junctions within this electron transport chain in non-cyclic electron flow, what's happening is that the energy that is released as these reduction reactions are happening is being used to take some hydrogen ions and to pump them into the lumen. And so what's happening as a result is that we're ending up with a buildup of hydrogen ions and thus an electrochemical gradient inside our thylakoid discs. Okay, so that is sort of our precursor to today's conversation. There's one more concept that I think is worthwhile us remembering, which is that what is the role of water? What does water do in the electron transport chain in non-cyclic electron flow? Well, I'm hoping you remember that in photosystem 2, in other words P680, that during photoexcitation we have electrons promoted and that are being grabbed onto by a primary electron acceptor. And depending on what photosystem, that primary electron acceptor changes. In the case of P680, we know that that is plastoquinone. But when those electrons leave, they need to be replenished or replaced. And so when we split water using a Z protein, then we produce some hydrogen ions, and we produce an oxygen, if we're looking at molar ratios, half of an oxygen molecule, but we also release a couple of electrons. And so that is the splitting of water is important because it helps to fill the hole in P680 so that photoexcitation can continue to happen and the electron transport chain can continue to be fueled. Okay, now we're ready to start chemiosmosis. So I'm hoping that you will find chemiosmosis during non-cyclic electron flow to feel very, very similar to the electron transport chain in chemiosmosis in cellular respiration. So there's a lot going on in this diagram. So I'm going to encourage you to pause the video and to take a look at what's going on here and try to make sense of all of the pieces before we move on and I start to talk about it. Okay, so let's orient ourselves a bit. So you can tell here, I'm hoping, from this particular shape that this is a phospholipid, and it's a phospholipid bilayer representing the thylakoid membrane. So as I sort of already mentioned today, we have an electron transport chain that is embedded in that thylakoid membrane. So all we're doing in this is that instead of the diagram that we looked at yesterday where it looked more like a graph, we're simply showing these electron transport chain complexes in their sequence, but also as embedded in the thylakoid membrane. So nothing should be new here. We have the stroma on the outside of our thylakoid, and we have our lumen inside the thylakoid. And what you should be seeing here is just the proper sequence of events in that electron transport chain. So we're going to move on past that because there was one important component of non-cyclic electron flow that relates to chemiosmosis, which is that for every 2 electrons moving through the electron transport chain, 4 hydrogen ions are pumped through into the lumen using our B6F complex. So as the electron transport chain continues and electrons continue to fill the hole in P680, we have this continual supply of protons or hydrogen ions filling up our lumen and creating that strong electrochemical gradient. Now, as you would expect, as we saw in cellular respiration, hydrogen ions can't just travel through our phospholipid bilayer because they're charged, and our phospholipid bilayer has these tails on the inside that are hydrocarbon tails, and so they're super hydrophobic. So those hydrogen ions are not going to naturally want to leak through our thylakoid membrane. So what happens is this buildup of hydrogens inside the lumen occurs, and the only way that they can pass through is through ATP synthase, which is the same enzyme that we saw in chemiosmosis in cellular respiration. So in cellular respiration, I talked about how this hydrogen ion buildup creates a bit of pressure that results in some energy being formed. And we thought about it sort of like if you had a garden hose. So garden hose, and water comes out the garden hose. But then what happens if you all of a sudden, you want to create pressure in your garden hose to wash something? Some of us will sort of create a kink. That's what we do at my house. We create a bit of a kink, and that creates a buildup of water here, and thus higher pressure coming out of that hose. So in other words, there's higher energy out of the hose. So we can sort of think about that with our hydrogen ions as they build up here and they have to get squeezed through ATPase. That just like in cellular respiration, create some proton motive force, and that force is used to phosphorylate ADP into becoming ATP, which then goes on to the Calvin cycle. Okay, so that's really all there is to chemiosmosis in photosynthesis. It's the same idea in principle, same mechanism, but with a few small changes in details. In cellular respiration, we would have seen the protons moving into the mitochondrial matrix and then, out of the mitochondrial matrix and then flooding back in. But in photosynthesis, we see our hydrogen ions being pumped into the lumen and then moving out through ATPase along with that proton gradient that's been created by our electron transport chain. All right, so let's write a little bit of this down, and I still have our diagram here for us to refer to if need be. Okay, so first things first, we have an electrochemical gradient that gets created as hydrogens are pumped into our lumen. Into that thylakoid space from our stroma. But they can't just move out anywhere that they want to. They have to move out through, they have to move out through ATPase because the thylakoid membrane is just not permeable to our hydrogen ions. And that generates that proton motive force that's going to phosphorylate ADP into ATP along so that it can be utilized to fuel the Calvin cycle. Now, we didn't mention this, but you have to remember also that NADPH from our electron transport chain, that final electron acceptor, is going to move on also to the Calvin cycle and help to fuel that process. Okay, so new term for us here, photophosphorylation. So the process of taking our ADP and phosphorylating it to ATP is called photophosphorylation. Why? Because it requires light. So if you hear that term, I think that's kind of makes sense, but now that we've talked about it, hopefully you'll remember that specialized term. Okay, now you should know the ratio of ATP to H+. We saw in cellular respiration that depending on whether we were talking about FAD or NADH or NAD+, the number of hydrogens would differ. So you need to know here that in cellular respiration, in photosynthesis, we're going to make 1 ATP for every 4 H+ as they pass through ATPase. Okay, so really important here, and you won't have much context for this yet because we've only talked about non-cyclic electron flow, but non-cyclic, implies that nothing is recycled, right? And so in this case, what might we recycle? Well, we might recycle an electron because that's what's moving through the chain. But in this case, it's called non-cyclic because we are not recycling that electron. It doesn't somehow get regenerated, and it becomes a part of that NADPH molecule. So this is a bit of a sneak preview to what we're going to be talking about when we discuss cyclic electron flow. So I want you to be making some predictions here. Okay, so short note on cyclic electron flow. So I want you to just pause this video, take a couple minutes to juxtapose what is in this diagram in comparison to what we saw with non-cyclic electron flow. Okay, so what I'm hoping that you were able to see was that we have cyclic electron flow is part of this larger non-cyclic electron flow. And so in the diagram, they've intentionally sort of highlighted one component while blurring a couple of others. So cyclic electron flow, when we're talking about it, what we're referring to as being recycled are electrons. So in non-cyclic electron flow, we have P680 promoting electrons being moved to fill the hole in P700, eventually being promoted and then becoming a part of the NADPH molecule. Well, cyclic electron flow, instead of the electrons, when P700 gets photoexcited and promotes electrons, instead of being transferred from ferredoxin to NADP reductase and being part of that NADPH molecule, they instead go from ferredoxin. Instead of going to the right, they go to the left. They go back to the B6F complex. And you'll remember from non-cyclic electron flow that B6F complex results in protons being pumped from the stroma into the lumen in order to create that electrochemical gradient, which inevitably results in ATP. So you might be wondering, why? Why the heck is this happening, and why is cyclic electron flow important? Well, what you haven't learned yet that we're going to be talking about very soon is that the Calvin cycle uses more ATP than it does NADPH. So you'll remember that, and so let me break this down. So if we have non-cyclic electron flow, we were producing 1 ATP and 1 NADPH per run-through of the cycle, of the non-cyclic electron transport chain, right? So for every 4 H pluses, we're producing 1 ATP, and for every 1 run of the electron transport chain, we're producing 1 NADPH. So this is the problem for the Calvin cycle. And we're about to see the Calvin cycle, but the problem is the Calvin cycle requires more ATP. The amount of ATP required is greater than the amount of NADPH. So if we're using more ATP than we are NADPH, and the non-cyclic electron flow is only going to produce a ratio of 1:1 for ATP and NADPH, we're going to run into a problem with the Calvin cycle. So what the function of cyclic electron flow is to produce extra ATP without producing extra NADPH, because that would be wasteful and a wasteful use of energy during the process of photosynthesis. So that's the gist of it. That's really, if we had to sum up, what is the importance of cyclic electron flow? What is its function? Well, its function is to recycle electrons in an effort to produce the additional ATP required to fuel the Calvin cycle, because it utilizes more ATP than it does NADPH. And that's it. So let's write a little bit down about this. Okay, so you'll remember that photosystem 1 is the only photosystem involved in cyclic electron flow. And so, that its electrons are going to get photoexcited, same as before, and they are still going to get transferred to ferredoxin, but instead they're being recycled back to B6F complex. Okay, so it's going to pass through to ferredoxin, and then back to that Q cycle with, plastoquinone, to B6F complex, and then back to chlorophyll P700, right? And so that's going to happen to produce as many ATP as are necessary to fuel the Calvin cycle. Okay, so as a result, we're not going to be producing additional NADPH because the electron never gets transferred to NADP reductase in cyclic electron flow. So no additional NADPH produced when we don't really need any more. Okay, so we're only going to be producing, we're going to be pumping extra hydrogen ions back into the lumen and therefore producing ATP by chemiosmosis without producing the additional NADPH. Okay, so that is the gist of cyclic electron flow. I hope I've outlined why it is necessary as an add-on to our light reactions. And so stay tuned, because our next conversation will center around how we're using our ATP and NADPH to fuel the Calvin cycle.